Hi everyone, we're ready to pick up where we left off in chapter 3. So I'm going to share my screen so we can go right to the PowerPoint. We're going to talk about section 3.4, the cell cycle. I'm going to begin by showing you this video. I think it does a good job sort of laying the groundwork for what we will talk in more detail about. So let me go ahead and have us watch that.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, Anaphase, and Telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Okay, that is a really excellent animation or video, and I would recommend, after we get done talking about the cell cycle, that you go back and watch that a second time, because it is really excellent and it does a super job in taking you through the different stages of the cell cycle. So we're going to go back to the PowerPoint and talk about this in, again, more detail. It's important that you understand that when we talk about this cell cycle, that this is taking place in what is referred to as somatic or non-sex cells. So for example, when you get a cut, that paper cut that you got on your finger after a couple of days heals, doesn't it? That's due to this process that is initiated upon tissue damage, or when you break a bone and you have it cast. After a few months, the bone is fully repaired. That's due to cell division that helps to reunite the broken ends of the bone. We'll be talking more about how this is regulated in section 3.5, which will be a separate Zoom lecture. So This process does not take place in sex cells. What are sex cells or gametes? Those are egg and sperm, okay? There's a different process that describes how they form. It's referred to as meiosis or meiosis. And we're not going to get into that in A&P, but maybe you've learned about it in biology. So this particular process taking place in somatic cells, you've seen this diagram in the video just a moment ago that sort of takes the cell cycle and divides it up into three major components. Listed here on the left-hand side. Interphase, which is the yellow, orange, and brown pieces of this big pizza. Followed by mitosis, shown here in green. Mitosis, as it mentions here, is division of the nuclear material. This is the DNA that helps make up the chromosomes that we'll talk more about. And finally, at the very sort of end here of mitosis is another process that's not very well delineated on the pizza, but it's mentioned here, cytokinesis. This is defined as division of the cytoplasm. This is what gives rise to the two daughter cells that are eventually formed. Remember, we start with what's called a parent cell that undergoes this process of the cell cycle. The end result is the formation of two daughter cells. This lower segment of the diagram here, where it talks about proceed to division, remain specialized, or die, we will talk more about that in the lecture regulation of the cell cycle in section 3.5. This is just another way of showing some of those different components of the cell cycle. But here we've introduced some sketches, right? As well as some diagrams that the artist has provided us to kind of tell us what's going on during different parts of interphase or mitosis. Here are those two daughter cells I was referring to a few moments ago. We start with a parent cell and we end with these two daughter cells. Keep in mind that these two daughter cells are genetically identical to one another. They're also identical to the parent cell that they were formed from. So I would suggest you come back to this particular diagram on occasion and review it, because it does a super great job in showing you again what's going on via the artist's sketch, as well as what's going on within the cells themselves. These are stained cells showing actual structures corresponding to the different stages. And what I want to do next is I want to take us through that entire process, starting with interphase, going through mitosis, and ending with cytokinesis and the formation of the two daughter cells. So here we are in the first part of the cell cycle. It's a big, big chunk of the cell cycle, actually. Upwards of 90% of the time a cell is in the cell cycle, it's in this particular component or stage called interphase. And many years ago, when I took biology, they used to define interphase as the resting phase. They no longer use that term because it's really just the opposite. It was called the resting phase because if you look at a cell under the microscope, there doesn't appear to be much going on. And so scientists thought, well, it's just resting. It's getting ready to undergo mitosis. But the reality is, once we had better technology, we could look into the cell with electron microscopes and really study what was going on metabolically. There's a heck of a lot taking place in interphase. It is anything but a resting stage. It's here that the cell is doing what that cell is supposed to do. A bone cell, a muscle cell, a nerve cell, a skin cell, a liver cell, a brain cell. Pick any cell you want. Most of the time, that cell is in the interphase stage. So it's contracting if it's a muscle cell. It's producing sweat if it's a cell of the sweat gland. It's providing rigidity if it's a bone cell. It's relaying impulses if it's a muscle cell, or I'm sorry, a nerve cell. So it's doing what it's designed to do during this interphase stage. However, if that cell is going to divide, it will take its DNA and replicate it here in interphase, because that replicated DNA will eventually be distributed to the daughter cells that are going to form at the end. So we're going to be talking in just a second about the replication of the DNA. This takes place in interphase. At the same time that the cell is undergoing this replication of the nuclear material, it's also got to get geared up to provide those daughter cells with adequate organelles, things like mitochondria, lysosomes, Golgi apparatus, rough and smooth endoplasmic reticulum, and so forth, things you learned about earlier in chapter 3, critical for the life of the cell. So these will also be replicated and more copies made, because again, those two daughter cells need their own lysosomes, Golgi bodies, smooth and rough ER, and so forth. They're also going to have their own cell membranes, aren't they? And the chemicals that are going to be required and needed for them to function as a muscle cell or nerve cell or a bone cell, a skin cell, whatever it might be. Those biochemicals are also produced during the cell cycle by the parent cell, all with the goal of producing two genetically identical daughter cells. So that big part of the cell cycle called interphase, which we saw earlier was the yellow, orange, and brown, I think, is divided up into 3 major subphases. And you see those listed here. The video talked about those. We have first G1, or gap 1, or you can think of it as growth 1, which leads us to S. This refers to the synthesis phase. This is when the DNA is replicated, followed by G2, gap 2, or growth 2. It's during G1 and G2 that the parent cell is making more organelles, making more membranes, making more biochemicals, gearing again up to give these to the 2 daughter cells that will be formed. The S phase, again, cannot stress this enough, the S phase of interphase is when the DNA replicates. So here we have a diagram showing the double helix of DNA, and we see it unwinding, and new daughter nucleotides come in and bond to the exposed bases of the parent strand. We're going to talk more about this coming up in chapter 4. But the end result is this initial double helix being replicated, if you will, to form 2 double helices down here, right? These are identical to one another. They're identical to the parent DNA. And again, these will be divvied to the 2 daughter cells eventually if we go through and follow the stages of mitosis, which we're going to do in just a few minutes. So interphase sets the stage. It really, really does. And when you look at a cell in late interphase, in G2, here's the artist sketch. Here's the actual photograph of a stained cell. This is, again, shown in one of the earlier slides. What do we have present? Well, we have a well-formed cell membrane or plasma membrane, which we talked about in the earlier lecture. Here it is, of course, in the actual stained cell. We have a well-defined nucleus. Can't hardly miss that, can you? Here's the cytoplasm that's containing all of our organelles. We're not going to really talk too much about those, but the mitochondria, lysosomes, Golgi body, and so forth, they're all in here. So the artist sketch illustrates the fact that that nucleus is surrounded by what's called a nuclear envelope. And within it is the DNA. That DNA is also associated with different kinds of proteins. They're called histone proteins. And together, the DNA and those histone proteins make up this loose strand of material called chromatin. A very, very important word to know. Chromatin is the DNA and associated proteins found here in the nucleus of the cell in late interphase. Here we also see another organelle that we're going to talk more about. They come in pairs. You see 2 pairs of these. They're called centrioles. You're going to see their importance in just a few moments. So we're setting the stage, if you will, now for a division of the nuclear material. But if you look at a cell under the microscope, it doesn't appear that a heck of a lot is going on. Hence why it used to be called the resting phase. It's not, remember, it's very, very active. It's growing, it's doing what the cell is designed to do. We now enter true mitosis, division of the nuclear material. There are 4 stages to mitosis: prophase, metaphase, anaphase, and telophase. This shows the first of the 4 phases, prophase. And note that we also have this term, late prophase. So we could actually have, in any of these 4 stages of mitosis, early, mid, and late components. Okay. How does this cell in prophase, either the sketch or the actual photograph of the stained cell, how does that differ from what we saw a moment ago in interphase? Well, here's some changes that are taking place in the cell. One of the most important things that takes place during prophase is the degradation or dissolving of the nuclear material. That's one thing that happens. And if you look inside, we don't have chromatin anymore. We have replicated chromosomes. Now there are 4 of them shown here, and they look like little kind of X-shaped structures. These 4 replicated chromosomes are each composed of 2 sister chromatids held together by what's called the centromere. So you see the sketch is pointing to one here, we have one here, we have one here, we have one here. What you really need to understand and remember is that within this replicated chromosome, or this one, or this one, or this one, we have the 2 sister chromatids. These 2 sister chromatids are genetically identical to one another. Just like these 2 sister chromatids, this one and this one are genetically identical to one another. The same applies to this guy. This chromatid is identical to its sister chromatid. And that's because of the replication of the DNA that took place back in the S phase of interphase. So the chromatin condenses and forms these replicated chromosomes. Note also that those pairs of centrioles that we saw earlier, they begin to migrate toward opposite ends of the cell, and between them form spindle fibers or microtubules. We don't see that so much in the photograph, but in the sketch in late prophase, those centriole pairs have moved toward opposite ends of the cell, and we see the development of what's called the spindle apparatus beginning to form in the core of the cell. And some of those spindle fibers are going to attach to the centromere regions of our replicated chromosomes and start to move them around a little bit. In fact, you might get a sense that in early prophase, they were sort of in this kind of central jumbled area of the former nucleus, and now they're starting to spread out, aren't they? They get spread out and they move apart thanks to the action of these contractile microtubules. In the next stage of mitosis, called metaphase, the replicated chromosomes have aligned themselves thanks to the action of the microtubules, the spindle fibers, in the central region here of the cell. This is sometimes called the metaphase plate. In the sketch, we see those 4 replicated chromosomes lined up, of course, from top to bottom. And in the actual stained cell, we can see those as these dark black stick-like structures. So those are more than just 4 chromosomes. These are probably, I would say, several dozen replicated chromosomes all lined up. You can see where the centriole pair would be one here. You can't see them, but that's where it is in the middle of this kind of pinkish, hazy stuff. And there'd be another centriole pair right here. This guy would be right about here. And then if you look super close, you can see these spindle fibers. You can actually see those. Those are these structures here. There are actually other spindle fibers that help attach these centriole pairs to the cell membrane, these little stubby guys in the sketch, but actually they can be quite extensive and elongated, like this actual photograph shows. The next stage of mitosis, called anaphase, is where we we divide the centromere, which remember was holding those sister chromatids together, and we now separate the sister chromatids. Once they separate, they're now called daughter chromosomes. It's kind of a funny term to use, I guess, but we don't call these sister chromatids anymore because they've separated from one another. These are now called sister chromosomes. Now remember, this sister chromosome, is identical to this sister chromosome. This one is identical to this one. Remember, they were held together in metaphase. And now we've pulled them apart. The spindle fibers contract, and these guys get pulled to this end, and these chromosomes, daughter chromosomes, get pulled toward the opposite end of the cell. Here we see that happening in the actual stained cell. These dark black structures are the sister chromosomes. They're getting pulled toward their centriole pair. These guys moving toward this guy, these guys moving toward this. This again is due to contraction of some of those microtubules, some of those spindle fibers. The cell may actually even begin to elongate a little bit during anaphase, especially late anaphase, which sets the stage for our last stage of mitosis called telophase. And the end of that process sort of merges with division of the cytoplasm called cytokinesis. Now, what makes telophase different from anaphase? Well, the major difference between stages 3 and 4 has to do with what eventually begins to form around those daughter chromosomes. We can begin to see in this artist sketch the reformation of nuclear envelopes. The cell, as I mentioned earlier, likely elongated a little bit too. And one thing we look for in late stages of telophase is the formation of what's called a cleavage furrow right down the center of the parent cell. You can see this sort of distinct line that's kind of forming here. It hasn't quite divided the cell in half yet. The cell is beginning to sort of pinch in half. So cytokinesis is beginning to take place. We're starting to reform nuclear envelopes around these daughter chromosomes. And eventually, at the end of telophase and at the end of cytokinesis, we will have taken that parent cell and divided it in half, thus forming the two daughter cells. Now remember, what do you know about their chromosome number, the daughter cells? The answer is they are genetically identical. So here is that process of cleavage furrow development in telophase. Once completed, the two daughter cells will be fully formed. We have reformed the nuclear envelopes, and the daughter chromosomes will begin to decondense back into chromatin. Remember, we talked about chromatin earlier. Chromatin was protein, those histone proteins, and DNA. So back to this kind of loose strandular look, if that's the word I can use. Loose strands of chromatin now form in the nucleus of those two daughter cells. And of course, we're left with a pair of centrioles too. We also are left with what we cannot see here in the cytoplasm, and that is new mitochondria, new lysosomes, new Golgi bodies, new smooth and rough ER, and so forth. So that's the end result. And look, we are back to interphase. We've made the entire loop from beginning to end. These next few slides just show some interesting photographs. These are scanning electron micrographs that are simply taking you through that cytokinesis process at the end of telophase, where we form that cleavage furrow, and here's our two daughter cells beginning to form. Very interesting process to watch in time lapse. By the way, this whole mechanism of cell division can take hours in some cells. So in review, here are the 4 stages of mitosis: prophase, metaphase, anaphase, telophase. You can go ahead and just review that. And then, of course, we have this sketch that we had a few moments ago earlier in the lecture. Now you can take a look at this and follow along from the very beginning of the process. Here's our parent cell in late interphase, and we go through the mitotic stages, eventually cytokinesis, and then here's our two genetically identical daughter cells. And that's the end of this lecture.